Well, this is our center, the JDRF Center of Excellence in New England, and I want to start with our team. It's a tremendous team, and I want you all to notice that I've listed them here in alphabetical order because there is no preeminent member. 80 lab members, 3 institutions. We have 3 basic themes. One is taking stem cell-derived islets and making them so that they evade immune and other stresses once transplanted. This whole concept got a huge shot in the arm with the cells that were made with Doug Melton's technology by Vertex and announced this summer at the ADA meeting in New Orleans. No one can say anymore that stem cell-derived islets can't restore insulin-independent euglycemia because it's been done. And you can't just say it's only one patient. It'd be like taking a dog on stage and having that dog talk and say, yeah, but it's only one dog. This establishes the new bar. We can make cells that restore insulin-dependent euglycemia. Now, the caveat, of course, is that this patient needed immunosuppression, and immunosuppression has its own toxicity. So the name of the game is, how are we going to make these cells so that they resist the stresses that will occur when they're transplanted into somebody? One of our strategies for identifying genes that we can knock down is done by Peng Yi and Stefan Kissler from Joslin. And what they've done is take NOD mouse beta cell tumor line NIT1 cells, generated random mutations in all the coding genes of those cells, and then put them in both an allogeneic and an autoimmune environment, and see which random mutations have made those cells resistant to alloimmune or autoimmune destruction. Now, there's several caveats with this approach. We think it's powerful and democratic, but it does assume that the mouse and human immune systems are similar, and we know they're not. And the NIT1 cells are tumor cells, so perhaps that will lead us astray as well. To get around that caveat, Judith Aguda in our group is creating ES cell lines from mice, both C57BL6 and the NOD, and then recapitulating the same differentiation protocol that Melton's lab figured out so that we can take more primary mouse SC islets and run them through the same screen I described on the previous slide. Now the question is, how do you test human cells other than putting them into a human? And for that, we rely heavily on work that Dale Greiner and Mike Brem have been spearheading here for the last 15 years, where you can take modified or unmodified SC human islets and then put them into a mouse, and then transfer in various components of the human immune system, both alloimmune and autoimmune, and see if those cells survive that environment. There are some caveats with this approach as well. It's time-consuming and expensive, and the mice can develop mouse graft versus host disease. And we know that the human immune system in the mouse isn't exactly like the human immune system in the human immune system. So for that, we relied very heavily on Lenny Schultz in the Jackson Laboratory, who was continuously improving our various mouse models. We can reduce the mouse graft versus host disease response by deleting the mouse MHC molecules and genetically alter the mice so that more native human immune system engrafts in those mice. And we have various diabetes models to induce diabetes to make sure that the human transplanted islets are what is restoring the mouse's euglycemia. Last but not least, Jason Gallia, Sally Kent, and Renee Marr are examining in granular detail the immune response that humans have against SC islets. Jason Gallia is testing techniques to randomly insert potential antigens that the human immune system is recognizing. And then Sally and Jason are studying the human immune system that infiltrates the human islets in the NOD SCID mouse recipients. And Renee Marr is doing single cell sequencing to analyze those cells. Since we recognize you might want to put some genes back into SC islets to protect them from the immune process, not just knocking out genes. All of the stuff that we do here is the long vision of Doug Melton, with support from the JDRF, obviously, and our main benefactor here, John Cammett. We are focused like a laser on coming up with a cell line that can be reasonably called the cure for type 1 diabetes, because it does survive transplant into individuals with diabetes.